Hello, this is a lesson on histology by Dr. Uma, courtesy of Medical Students Arena. Today we are going to have a study of tissues, that is histology, and we are going to embed on what you are able to see from your hand. We are going to check on the primary tissue classes, embryonic tissues, interpreting tissue section, epithelial tissues, simple epithelia, and stratified epithelia. This knowledge is basically for your good and grasp in your medical career and the entire journey as a doctor. So primarily, I will start by saying it's like 50 trillion cells and thousands of organs that exist in the human body. They may seem to be a structure of forbidding complexity, but sometimes, fortunately for her health, or maybe what we refer to as the longevity and self-understanding, the biologists of a past generation were not discouraged by this complexity, but discovered patterns that made it more understandable. So one of these patterns is the fact that these trillions of cells belong to only 200 different types or so. These cells are organized into tissue that fall into just four broad categories. Tissues that falls into that four broad categories. That is epithelial tissue, connective tissue, nervous tissue, and muscular tissue. So an organ is a structure with discrete boundary that is composed of two or more of these tissue types, usually maybe four. You see, an organ can have both connective tissue, epithelial tissue, nervous tissue, and muscular tissue. So that is a complete organ. And as you all know that from the primary knowledge and what we obtained from form 2 biology class, that a cell, maybe form 1, let me relate it back to form 1, is that a cell has got cell organelles, and the cell organelles have got molecules, and the molecules have got atoms. So all this hierarchy comes into when we have a cell, and then cell will merge as much as they're typically and similar in function, they'll give us what is referred to as tissue. Then more than one tissue with structural similarity and functionality, they'll give us what we refer to as organ. Here we are in organs. So organs will derive their function not from their cells alone, but from how the cells are organized into tissues. So cells are normally at all, they're specialized for certain tasks like muscle contraction, defense, enzyme secretion, and so forth. So no one cell type has the mechanism to carry out all of the body's vital function alone. Therefore, they will work together at certain tasks and form tissues that carry out particular function. For example, the nerve now signal or nutrient digestion by the digestive system. That is now an entire organ system. I hope you are familiarized with that. And so let us go on. I'm very sure that I'm not at high speed and you are able to see because the chapters are kind of outlined in a very discreet way. So I want to talk about what we refer to as the study of tissues now in details. How they are arranged into organ is what we call the histology, or what we can refer to as well as microscopic anatomy. I want you to know the difference between microscopic anatomy and gross anatomy. Gross anatomy, they don't entail miniature or small particles, but they entail objective-oriented things which you can view with your naked eyes. So when you talk about histology, they are the smallest one, the microscopic anatomy. And that is the subject of this entire content that I want to give you. Here we're going to study the four tissue classes, the variation within each class, and how to recognize tissue types microscopically and relate their microscopic anatomy to their function, how tissues are arranged to form an organ, right? And how tissues changes over the life of the individual as they grow, shrink, and change from one tissue type to another, that is, okay? So, like for example, histology will bridge the gap between the cytology of the preceding chapters and the organ system approach of the chapters that we are going to check in the next. Now, let's start with the study of the tissue. And I want you to understand that our objective today is that we're going to name four primary classes of tissues into which all the adult tissues are classified. As well, we are going to name the three embryonic germ layers and some adult tissues derived from each. And again, we're going to visualize the three-dimensional shape of a structure from a two-dimensional tissue section. So starting from the first thing that we are able to see, what we refer to as the primary tissue classes. So first of all, I know you know a tissue is a group of similar cells and cell products that arise from the same region of the embryo, and they will work together to perform a specific structural or physiological role. that is in an organ. So the four primary tissues are the epithelial, the way I said initially, epithelial, connective tissue, nervous, and muscular tissue. These tissues differ from each other in the types and function of their cells. So the characteristic of the matrix, and what is matrix? The extracellular materials is also different. So when I say matrix, I want you to think like that is what surrounds the cells, okay? And the relative amount of space occupied by the cells versus matrix. Like, I want you to know your body, cells are there, organs are there, and tissues are there. But between the spaces, between the cell and the cell, that entire thing is matrix. So when I say matrix, just like a mesh up, which is filling up the space between those things. So the cells are so close together that the matrix is now scarcely visible. While in connective tissue, that is another type of tissue which I already mentioned, the matrix usually will occupy much more space than the cells do. So like the matrix is now composed of fibrous protein that I don't want you to forget, and usually a clear gel variance known as what we refer to as ground substance. And you need to know the ground substances so that you are able to know the kind of component, the ground substance, and when you are doing administration of various injection like intramuscular injection, you know when you're doing that, what comes forth. So when I say ground substances, they are what now give us the matrix, and the component of matrix is the ground substances, tissue fluid, and extracellular fluid, and maybe interstitial fluid, or what we refer to as the tissue gel. Now, in cartilage and bone, it can be rubbery or stony in consistency. So the ground substance contains water, gases, minerals, nutrients, waste, and other chemicals. But what I want you to know in general that a tissue is composed of cells and a matrix, and the matrix is composed of fiber and ground substances. Simple like that. So let's go to the next subtopic, that is embryonic tissue. Now, let us see, when you talk about embryo, that is where the entire thing comes from. I mean, during the organogenesis, the embryogenesis, I know you know, we have zygote, after fertilization we have zygote, then embryo. And this is a developmental milestone within the uterus, or what we refer to as this intrauterine developmental process. So we want to see where tissue comes from. That is why we want to discuss embryonic tissues. Okay. So human, we know they develop. The development of human begins with a single cell, right? They fertilize the egg, which soon divides to produce, yeah, it produces things to do with, it produces cores of identical smaller cells. They divide, they keep on gawana, right? So the first tissue appears when these cells start to organize themselves into layers. First, two, and soon three strata called the primary germ layers, which now will give rise to all of the body maturity. So when I say primary germ layers, these are the beginning layers of the fundamental unit of tissue. And the three layers are what is ectoderm, that is one, mesoderm, and endoderm. Now for your exam purpose, you need to know which tissue comes from ectoderm, which tissue comes from mesoderm, and which tissue comes from endoderm. So that is what I want to just give you brief. Now, from your analysis, just ectoderm is outside, endoderm inside, and the mesoderm is in the middle. So I want you to understand this way. The ectoderm is an outer layer that gives rise to things which are outside the body. That's like epidermis and nervous system. The inner layer, the endoderm, will now give rise to mucous membrane of the digestive and respiratory tracts and to the digestive gland as well, among other things also which are there. Then between the two, that is between the endoderm and ectoderm, we have the mesoderm. And the mesoderm is a layer of more loosely organized cells, and the mesoderm will eventually turn to what we refer to as the gelatinous tissue called mesenchyme. And this mesenchyme is composed of what I can call them fine wispy collagen fibers like or branching cells, right? Which we can call the fibroblast. So at most, fibroblasts found in the connective tissue. So nikisema hivi, just calculate that. Therefore, the mesoderm give us the connective tissue in general. So the mesoderm give us mesenchyme, which mesenchyme will now give us connective tissue, right? So it gives rise to connective. An example of connective tissues are like the muscles, the bones, and the blood, among other tissues which are found there. So that is that. So the next subtopic is interpreting tissue section, epithelial tissue, and then you see the simple epithelium, stratified epithelium, then we call it session, we go to the next part of histology. Now, let's see how we can be able to interpret tissue sections. Histologists use a variety of techniques for preserving, sectioning, and staining tissue to show their structural details as clearly as possible. So tissue specimens are preserved in what we refer to as fixative. And I want you to understand the difference between fixative and fixation. I had told you previously in the other class that fixative is a chemical which is used to preserve a specimen or a section of a tissue to prevent it from decaying. So when I say fixative, talk about chemical. And then fixation is now the process of applying fixative to a tissue to prevent it from decaying. Now, what is a fixative? A fixative is that chemical. So for example, we have formalin in the most of the morgue and the hospital, which are used to preserve dead bodies in the morgue, that is mortuary. So I wanted to know that after fixation, most tissues are cut into thin slices called histological section. And about the section, you've already known about this in previous classes, just in your high school career, course line. So these sections are typically only 1 or 2 cell thick. They must be thin so that they can allow light to penetrate through them. So they will allow the light of microscope to pass through and to reduce the confusion of the image that will result from many layers of overlapping cells. So they are mounted on slides, yeah, and after that they are artificially stained or colored with histological stains. For example, I know you know about the ethanol, the blah blah blah, methyl blue, and so on and so forth. So yeah, so why do we stain them? To bring out a detail when we view them using microscope. You know, various parts of the cells, like some organelles, they will give a different color depending on which stain. So at least we are able to distinguish nucleolus from the ribosomes, from the lysosome, and from the Golgi apparatus. So that is what is importance of stain is. So if they were not stained, most tissue will appear very pale gray with maybe stains that bind to different components of the tissue. So, however, you may see pink cytoplasm, maybe violet nuclear, maybe blue, green, or golden brown protein fibers, depending on the stain used. Sectioning a tissue reduces three-dimensional structure. So at least we can have two-dimensional slice. So you must keep this in mind and try to translate the microscopic image into a mental image of the whole structure. So don't think that what you are seeing there is everything. By cutting it or sectioning it, you have reduced the three-dimensional structure to two-dimensional structure. So like the boiled egg and the elbow macaroni, you can see they can be looking so quite different when it is cut at various different levels or planes of section, right? Just similarly, like a cold tube such as gland or the uterus is often broken up into multiple portions since it meanders in and out of the plane of section. And you see this, that. So I need to note that something like a grazing slice through a boiled, let me say a boiled egg might miss the yolk when you cut it or section it. So if you are viewing it, figure out where will you expect the egg to be. So just as a tissue section might miss the nucleus or cells or any other thing in the ovary, even though this structure were present, so you need to picture it. I want to drop in an image which I'll delete immediately so that at least you can be able to see what I'm talking about. So what you are seeing from your hand, example of some sectioning, and I hope you're able to see what I'm talking about. I know it's not that big, but I can make it big to some extent so that at least you can be able to see the kind of section I am talking about. So we have longitudinal section, cross section, oblique section, depending on how you're sectioning. And the plane you did in introduction of anatomy, so this is not a problem to you. So let's proceed where we were. So another thing which is very important that many anatomical structures are significantly longer in one direction than other. So for example, the humerus and esophagus, for example, they are very longer in one dimension, maybe longer posterior or anteriorly or superiorly than posteriorly. So when a tissue is cut in a long direction, it is called longitudinal. So sema umeikata katikati kiteremka, that is longitudinal section. And one cut perpendicularly to this cross section, we call it now cross section or transverse section. Another section can be cut at an angle between longitudinal and cross section. That is what we call oblique section, and so on and so forth. So before you go on, I want to have some things in your mind, like I need you to be able to classify each of the following, like four primary tissue classes. Like you need to know, where do you find skin, where does skin falls in, like skin surface. Yeah, so I want to classify each of the following into one of the four primary tissue classes. Yeah, that is skin surface, fat, the spinal cord, most are tissues, bones, tendons, blood, the inner lining of the stomach. Where do they fall? I just want you to think of that. So from there, I also want you to, what are tissues composed of in addition to cells, right? What embryonic germ layers give rise to nervous tissues? Yeah, I want you to go and check on that. Again, I need you to go and check on what is the term for a thin stained slice of tissue mount, which are a tissue mounted on a microscope slide. What is the term for that? For a thin stains? Yeah, think about that. I've just said them right now. The next and the second last part of our lesson is that we're going to check on the epithelial tissue. And in epithelial tissue, I want you to have the following objectives. First of all, you need to be in position to describe the properties that distinguish epithelial from other tissue classes. You also need to be able to list and classify 8 types of epithelium, right? 8 types of epithelium. You also need to distinguish them from each other and state where each type can be found in your body. Again, I needed to be able to explain how the structural differences between epithelial relate to the functional differences. And I also needed to be able to visually recognize each epithelial type from specimen or photographs. Away from that, let's now start from epithelial tissue. I want you to know that epithelial tissue, among the 4 tissue, which I had already told you before, is that it consists of a flat sheet of closely adhering cells, one or more cells thick, with the upper surface usually exposed to the environment or to an internal space in the body. So epithelial covers the body surface, it lines the body cavities, forms the external and internal linings of many organs, and constitutes most gland tissues. The extracellular material is so thin it is not visible with a light microscope at all, and the epithelial will allow no room for blood vessels. So they do, however, almost always lie on a layer of a loose connective tissue and depend on its blood vessel for nourishment and waste removal. Now, between an epithelium and the underlying connective tissue, there's a layer called the basement membrane, usually too thin to be visible with a light microscope. So it contains collagen, adhesive glycoproteins. I'm talking about the basement membrane. What is the component? I've said it has collagen, adhesive glycoproteins, which we can call the lamina or the laminin, and fibronectin. And it also has a large protein carbohydrate complex called heparin sulfate. It gradually blends with collagenous and reticular fibers on the connective tissue side. And so the basement membrane serves to anchor an epithelium to the connective tissue below it. I want you to remember that the surface of an epithelial cell that faces this basement membrane that I've just said, it is what we call the basal surface. And the one that faces away from the basement membrane is what we call the apical surface. I wanted to picture what I'm talking. Is that just like epithelial tissue embedded imelala kwa basement membrane. So kuna part yake ambayo ina face basement membrane ambayo tumeita basal surface, na kuna ile ina face away from the basal membrane inaitwa apical surface. Hakuna vile epithelial itakaa peke yake kwa hewa, so that is what is important. So it has to lie on the basement membrane. Yeah, so between the epithelium and other connective tissue, we have that basement membrane layer. Now, let us classify epithelial because there are some classification of epithelial tissue. Epithelial are classified into 2 broad categories, either simple or stratified. When you talk about stratified, you know the layer. So if stratified has got layers, then simple doesn't have layers. So that is the main category, the simple and stratified. With the 4 types in each category, in a simple epithelium, every cell touches the basement membrane. Whereas in a stratified epithelium, when it is stratified, some cells rest on the top of other cells. Because ni layer nyingi nyingi, zile zimelala kwa ingine hazitaguza base membrane. So those ones who are lying on the top or resting on the top of the other layers or cells, they will not have the direct contact to the basement membrane. That is that. I'm going to share to you a given picture again, and I want you to be in position to at least see what I'm talking about. It is small, kind of small, but I'm making it big. Now this is simple epithelium, then we have stratified epithelium. What is down there is what we refer to as the basement membrane. So stratified, you can see there are more than one layer, so they're lying on the top of each other. And then simple epithelium is just one cell layer, and therefore all of them are directly into basement membrane. As you can see in stratified epithelium, most of them are floating suspended with the other layer, so they don't have direct contact with the basement membrane. Let us proceed. Let's start with one, simple epithelium, because I said we classified them either in simple epithelium or stratified epithelium. So when you talk about simple epithelium, generally simple has only one layer of cells, although this is a somewhat doubtable point in the pseudo-stratified type. So in the the pseudo-stratified type, when I say pseudo, is something which is false impressive, to give us false impressive. So I've said simple epithelium, they always have one layer. But when you talk about the pseudo-stratified type of simple epithelium, it appears to be having one layer, but or maybe more layers, but it is generally having one. So in simple epithelium, we have three types of simple epithelium, and they are named for the shape of their cells. So we have simple squamous, which is because of the thin scaly cells. We have simple cuboidal because of the square or round cells, and we have simple columnar because of tall, narrow cells. Cuboidal can be square, so don't, because the name is cube, don't just mean it square. Some of them look round when you initially take that perspective and use microscope to view them by yourself. And then we have simple columnar. They are ever tall, narrow cells. So in the fourth type, pseudostratified columnar, not all cells, when I say the pseudostratified columnar, not all cells reach the free surface. So the shorter the cells are, the ones which are shorter, they are covered over the taller ones. So this epithelium looks stratified in most tissue sections. But carefully when you examine them, that is in pseudostratified, the way I told you, it gives a false impression, especially when you use electron microscope. It will show that every cells reach the basement membrane. So simple columnar and pseudostratified columnar epithelium often produce a protective mucus coatings over the mucous membrane. So the mucus secreted by something like wine glass shaped goblet cells. So most of them, these two, their main purpose is to secrete mucus, because they have the goblet cells. So that is about the simple epithelial. I've talked about the simple squamous, simple cuboidal epithelium, simple columnar, and pseudo-stratified columnar that I said it can give you a false impression because of how it looks like. So let's go to the last part, that is stratified epithelial, from what you are able to see. Now in the stratified epithelium, we are going to talk about this. Now, in stratified epithelia, it will range from 2 to 20 or more layers of cells. And some of the cells, they will be resting directly on others, and only the deepest layer resting on the basement membranes. Three of the stratified epithelia are always named for the shapes of their surface cell as well, the way we mentioned priorly with the simple epithelia. So the first one is stratified squamous. I had already mentioned what is squamous for. Stratified cuboidal, stratified columnar epithelium. So the deeper cells, however, may be of a different shape than the surface cells. So the fourth type of stratified epithelial is what we call transitional epithelia. So apart from stratified squamous, stratified cuboidal, and stratified columnar epithelia, we still have transitional epithelia. And this name was named when it was thought to present or to represent a transitional stage between stratified squamous and stratified columnar epithelium. So this is how now it is known to be untrue, but the name has persisted up to now. So let us proceed. We are talking about tissues of the body, so don't be scared. Yeah, so if this is your primary class, I know medicine entails a lot. So let's proceed. Stratified columnar epithelia is normally very rare and you cannot see it easily. It's only seen in places where two other epithelial types meet. As for example, it is limited only to the regions of pharynx, larynx, anal canal, and male urethra. We will not consider this type any further. So other three types are illustrated and summarized. So we are going to have them and check on them later. So the most widespread epithelium in the body is what we refer to as the stratified squamous epithelium. And this one, it deserves further to be discussed, and therefore we'll give it time to prepare time to ongele. Even the deepest layer of cells, and they are cuboidal to columnar, and they normally undergo continual mitosis. So when we have layers of cells, the ones which are the lower region, they keep on dividing. That is what I say, it continues with the mitosis process. So they will form their daughter cells, which now will push toward the surface and become flatter, and then they will form the topper layer of the strata. So as they migrate further upward until they finally will die and flake off. And what did he say is flaking off? Just like exfoliating, yeah? So their separation from the surface is called exfoliation or desquamation. And therefore, that's what brings on for us the study of exfoliated cell, which is called exfoliate cytology, the way I told you before. So you can easily study exfoliated cells by scraping your gums with a toothpick, maybe smearing this material on a slide and staining it. And a similar procedure is used in the pap smear, an examination of exfoliated cells from the cervix for signs of uterine cancer. Now, stratified squamous epithelial are of two kinds. They can either be keratinized or non-keratinized. Keratinized epithelium, they are found on the skin surface, that is epidermis, and is covered with a layer of compact dead squamous cells. And these cells are packed with a durable protein keratin and coated with water repellent. So the skin surface is therefore relatively dry, and it retards water loss from the body, and it resists penetration by disease organism, and that is that. So when I say keratin, is also the protein of which animals' horns are made of. That is where it obtained its name keratin. Now, things to do with the tongue, oral mucosa, esophagus, vagina, and few other internal membranes are covered with non-keratinized type. So they lack the surface layer of dead cells. So this type, they will provide surface that is again abrasion resistant. So they will not allow any abrasion kugwaro, but also moist and slippery. So these characteristics are well suited to resist stress produced by either in your oral cavity or oral whatever. They will be reducing the stress produced by chewing and swallowing of food. And in your vagina, they will reduce the stress which is given by sexual intercourse and childbirth. That was just an epic, and we are just done with that point of the tissue. So we have several tissue type, yeah, and you really need to know which one is which one, right? And you really need to know what is exfoliated. Like now, this image I'm going to show you right now is for exfoliated tissue, which is under the these are exfoliated of squamous tissue. So I want to see it clearly from your hand before we go to the other class. You can see it has like exfoliated of squamous cells from the mucosal surface of the vagina. So this is that a photo by the G. Gesses and R. H. Cardon, tissue of an organ. So you can check this on a textbook of Atlas of scanning election microscopy. So aside from the gums and vagina, you can also name other epithelium in the body that will look like this to the scanning electron microscope where it is used. Again, I can show you one or more two tissues so that you know them, so you can identify them. At least let me show you a squamous epithelial tissue and you see how it looks like. This is what is squamous epithelial tissue. This is the external surface of a small intestine actually. I hope you are able to see it from your hand. That is it. That is it. From your hand, you're able to see that is a small intestine, simple squamous epithelium. So not much to be shown. There are very many of such, and you can follow me so that I can share image or I share it to the atlas or maybe a direct on where to obtain it. Our lesson is done. We are going to see on the next topic, and the following are what we are going to have in our histology next class. Be present so that you don't miss what you're seeing there. Connective tissue, and we're going to talk about the connective tissue overview, fibrous connective tissue, cartilage, bone, and blood. Thank you so much for attending. This is Medical Students Arena. You're always invited, and you can watch more of such in our YouTube channel. Please don't forget to subscribe.